This is Bio 101, Chapter 17, From Gene to Protein. In this chapter, we're going to be talking about the process of gene expression, which is how we go from a gene that is present on a piece of DNA to the formation of a protein, which is how a trait is actually expressed. Now, we're going to see that there are some similarities between the processes that occur in prokaryotic and eukaryotic cells, but there are a fair number of differences as well because prokaryotic cells lack a nucleus. And our attention is really going to be focused on the process in eukaryotic cells. So let's talk about the central dogma of biology, which encompasses all of these ideas about gene expression. When we look at the word central dogma, central means in, at, or near the center, and dogma is a set of beliefs or principles that are guiding within a larger idea. And so when we look at the central dogma of biology, we're looking at that set of principles that's really at the heart of everything we know about biology. And so this is kind of a little sketch of what the central dogma looks like. There are three types of molecules that are present here: DNA, RNA, and specifically this would be mRNA. We'll talk about the differences in a moment. And protein. Those are our three molecules that are present. There are also three processes present: replication, transcription, and translation. And so if we look at what's going on in this diagram, we see that DNA has the ability to make copies of itself through the process of replication. That's what this arrow means: DNA to DNA, DNA to DNA. So the DNA makes copies of itself through replication. That's what we talked about in our previous chapter. This process is only going to occur in S phase of interphase before those cells get ready to divide. Now, mRNA, or messenger RNA, is an RNA copy of a single gene on our DNA. And that RNA copy gets made through the process of transcription. So this is one step in gene expression, where we take that gene and we express it into an RNA copy. And in most cases, in order for us to see an effect on the expression of that gene, that RNA will then be used in order to build a protein in the process of translation. If we look at those words, transcript and translate, transcript really relates to the verb transcribe. And when we transcribe something, we're going to be changing something from one version of a language to another version of the same language. So for example, a transcriptionist is a person who listens to someone's verbal notes and transcribes them into written notes, writes them down or types them. But it's all the same language. And DNA and RNA are both nucleic acids, so we're seeing that we are transcribing from one version of that language into another. We're just making a copy. However, translation usually means that we're changing from one language into another language. And that's what we see happening here. RNA, which is a nucleic acid, gets changed into a protein or amino acid based language. So we're going to use this code as the basis for building this protein in order to translate from this to this. And the protein is actually the key, because this is what gives us a certain trait or a certain phenotype that is expressed from that gene. So let's look at these three players in just a little more detail. And you should remember a fair amount of detail up to this point because we have covered DNA, RNA, and proteins previously. DNA stands for deoxyribonucleic acid, and it is our double helix molecule. So it contains two strands that are complementary to each other. So adenine always pairs with thymine on the opposite strand. Guanine always pairs with cytosine on the opposite strand. The two strands are held together by hydrogen bonds, and one long strand of DNA forms this double helix and wraps up in around histones in order to condense and form a chromosome. One short sequence of that DNA is what we would call a gene, and that one gene codes for a protein. On a chromosome, we see that there are hundreds of genes, and in some cases thousands of genes that are present that code for different proteins. RNA is the second molecule that we're going to look at, and there are actually three different types of RNA. RNA itself stands for ribonucleic acid, so it is similar to DNA, but remember it has a different sugar, and it also is a single-stranded molecule rather than a double-stranded molecule. We still see complementary base pairing though in our RNA, except that adenine is going to pair with uracil instead of thymine because thymine is not present in our RNA molecules, but guanine and cytosine still pair. So when we look at an mRNA or a messenger RNA, this is going to be the RNA that is a copy of a gene and carries the message from that gene from the DNA out into the cytoplasm in order to build the protein. We also see tRNAs or transfer RNAs, and this is one long RNA molecule. You can see here I'm tracing it. But parts of that RNA molecule are complementary, which allows for this part, this part, this part, and this part to stick to each other to give us that sort of a T shape. T actually stands for transfer, and what we're going to see that the tRNAs do is they transfer amino acids to the ribosome as they're building the protein. rRNAs, or ribosomal RNAs, are very large, complex structures, and they are going to be partially responsible for building the ribosome structure itself. So ribosomes are composed of both rRNA and some protein to build up that structure. Finally, we see that proteins are the last type of molecule that are part of the central dogma, and we've talked about proteins before as well. Proteins are the verbs of the cell. They do a lot of things, and because they serve so many functions in our bodies, they are absolutely essential. We would not be able to survive without functional proteins in our cells. So let's kind of take a look at these processes. Now we've already looked at replication, so we don't need to spend any time on that today, but do remember that replication is how we make a copy of an entire chromosome, and that this is going to happen in S phase of interphase before a cell goes through mitosis or meiosis. The other two processes that we're going to talk about today are transcription and translation. These two processes will occur back to back in the process of gene expression. So they'll be happening all the time in the life of the cell. Depending on what's going on, a gene may need to be transcribed and then translated in order to make the protein. And various genes can be going through transcription and translation at various points in time. So it's one of those things that's kind of always happening in the cell, whereas replication only happens in a set point in time. Transcription does occur in the nucleus of the cell, and this makes sense because what we're doing is making a copy of part of the DNA. And because the DNA is housed in the nucleus, it makes sense that transcription would occur in the nucleus. And what we're doing in transcription is making an mRNA copy of the gene. And so what happens is we have a three-step process: initiation, elongation, and termination. So initiation means to start, elongation means to make longer, and termination means to stop. So in initiation, what we see happening is that a molecule called RNA polymerase binds to the DNA. The RNA polymerase is going to be very similar to the DNA polymerase that we saw playing a role in replication, except that DNA polymerase builds DNA and RNA polymerase builds RNA. And RNA polymerase functions in much the same way in that it's only going to build new RNA in the 5' to 3' direction. And so the RNA polymerase has the ability to bind to the DNA, and it's going to bind to a specific region called the promoter. So the promoter is a region before the gene actually starts, where the sequence of the DNA pulls in all these proteins called transcription factors and actually recruits the RNA polymerase with those transcription factors. So there's a certain sequence, including the TATA box, in the promoter that help to recruit all of those proteins to get this process started. Along with RNA polymerase binding, we also see that DNA unzips, and there is going to be a helicase involved in that process. In the second step, elongation, we're going to see that RNA polymerase is going to move along the length of our DNA strand, and it's going to build RNA as it moves along. And that RNA is going to be complementary to the DNA strand. So you can see here, this is the RNA transcript that has already been built, and as that RNA polymerase continues to move forward, it will continue to build a complementary strand. So where we see an A on our template strand, we have a U on our RNA transcript strand. And that's because we do not see thymine in RNA. Instead, we see uracil, and that's the complementary base. However, our thymine is still present in our DNA, and so the complementary base pairing rules of T to A would still apply here. So on the RNA strand, we see an adenine present at that spot. Where we see a guanine, we add a cytosine, and where we see a cytosine, we add a guanine. And so our RNA would be built by RNA polymerase moving along the DNA strand. Eventually, that RNA polymerase encounters a termination sequence, which is just a sequence of DNA that indicates that that's the end of the gene, and the RNA polymerase stops transcribing and falls off of the DNA. This allows the DNA strands to now go back together, releasing the mRNA, which is going to go through a process of modification and then get sent from the nucleus out into the cytoplasm. Okay, so that is the process of transcription. It's actually fairly simple. We see some of the same components as we saw with replication, and the DNA has to unzip. We have a polymerase that's going to build our new molecule, but here it's an RNA polymerase moving in the 5' to 3' direction, but it's only going to build a short sequence of the DNA, okay, a copy, a short sequence of the DNA, and then the original DNA strands will go back together. So if we were to look at transcription in action, here is our gene that we'd like to transcribe. So the first thing that we have to do is we have to separate those two strands. Then we have to know which strand we're going to actually transcribe. And again, there would be a promoter present that would indicate to the proteins which strand needs to be transcribed. For our quick little simple example, we're going to transcribe the bottom strand. And then we're going to start adding mRNA bases using uracil instead of thymine. So where we see an A, we're going to add a U. Where we see a G, we add a C. C, A, U, G, A, U, A, C, U, U, G, A, A. We've now built our mRNA strand, so that's a copy of that gene. And so what's going to happen is that the mRNA strand will end up leaving the nucleus and allow those two strands of DNA go back together, even though that's not indicated in our animation. Okay. Before our mRNA actually gets to leave the nucleus, it does go through a process or go through a modification process. We call it RNA processing. There are three things that happen. A 5' cap gets added to the 5' end. And when we say cap, really this is just a modified base. So instead of a guanine, it's a slightly modified version of guanine that gets added to the 5' end. And then at the other end, the 3' end, we're going to add a poly A tail. Poly means many, so we're going to add many adenines to the 3' end. Both of these are going to be involved in getting the mRNA out of the nucleus, but also the poly A tail helps to protect the mRNA from degradation. So as it's going through that process of recruiting a ribosome to start building the protein, it's vulnerable to being broken down by nucleases in the cell, and so that poly A tail helps to protect the actual message from being destroyed by those nucleases. And then RNA splicing is the third thing that happens in this modification process. Now when we say the word splice, sometimes we think about wires, and if you splice two wires, you're actually going to cut them and then reattach them in a different way. And so when we splice RNA, we actually end up cutting the RNA molecule in order to take out parts of the molecule called introns. So along the length of a transcribed mRNA molecule, there will be exons, which are going to be parts of that gene that actually code for protein, and introns, which are non-coding parts of a gene. And we want to make sure that we remove those introns before we actually start trying to turn that mRNA translated into protein, so that we actually translate the correct message. So that is RNA processing. Then we'll see that our mRNA moves on into the cytoplasm where translation occurs. And translation is the process of actually making the protein using the mRNA code. Now, one thing that's a little bit difficult to deal with is that there are about 20 commonly occurring amino acids present in the human body and really in nature. There are some that are kind of rare, but we're going to focus on those 20 commonly occurring amino acids. There are only 4 nucleotide bases in our mRNA. So it's not possible to make a direct correlation that every time you have an adenine, then you're going to add an amino acid of phenylalanine to your chain. And every time you have a guanine, you're going to add a proline. We can't make that direct correlation because there's only 4 bases and we have more than 4 amino acids. And so what we end up doing is that instead of reading our mRNA 1 base at a time, we read it 3 bases at a time. So these 3 bases, U, G, G, make up a single codon. And that one codon gives us the code that we need to determine the first amino acid in our protein chain. So the first amino acid is tryptophan. TRP is tryptophan. The second codon is UUU, which tells us that the second amino acid that we need is phenylalanine. So as we move along the length of the mRNA, we're going to be reading that mRNA sequence 3 bases at a time, and those 3 bases at a time are called codons. And this is a chart showing us how we would determine the amino acid that a codon codes for. So for example, if we have the codon ACG, our first base is always going to be over here on the left side, our second base across the top, and our third base over here on the right. And so for A, here's my first base is A. So I'm going to go across until I find the column that corresponds to the second base of C. Okay, so this is where A and C intersect. And so now I'm going to find in my list G as the third base. So ACG codes for the amino acid threonine. Okay, that's how we would use this chart. Notice that threonine actually is coded for by anything that starts with AC. So ACU, ACC, ACA, ACG. We call that redundancy in our genetic code, and it actually ends up being a good thing because, as we'll see later, if we have a mutation in that third base, it doesn't really cause any problems. So let's look now at the major players in this process of translation. We already know that the mRNA is our messenger RNA, which is a copy of our gene, and it's going to give us the code and codons that we need in order to build our protein. The ribosome is where this process of translation actually occurs, so this is going to be the site where we actually build the protein, and then the tRNA is the molecule that brings the amino acids in. It actually delivers the materials that we need in order to build the protein. So this is going to show us what a ribosome looks like. And ribosomes are actually 2 subunits. We have a large subunit and a small subunit that come together whenever they bind to an mRNA. In the large subunit, we see that there's an E site, a P site, and an A site. Okay, the E site is our exit site. So this is going to be where the tRNA goes whenever it's getting ready to leave the ribosome. Okay. The P site and the A site will hold tRNAs in order to form a bond between the amino acids that they carry. So you can see here, this is a tRNA, this is a tRNA. These are the 2 amino acids, and those 2 amino acids are going to form a bond, so the ribosome will form a bond between them. We can see that from the E site we've had a tRNA that has left. Okay. But these 2 tRNAs are still forming a bond between their amino acids. So we'll walk through this process, but this is essentially how it's going to be laid out. We'll have tRNAs in the P site and the A site. They form a bond between their 2 amino acids, and then the ribosome moves down, which shifts this tRNA into the E site. This tRNA gets shifted into the P site, and now the A site is open and it will be positioned over this next codon. And so the next tRNA comes in that matches up with this codon and carries the next amino acid, which allows us to form a bond and then the process repeats. Okay. This is what a tRNA looks like. We've already kind of mentioned this. The important parts of a tRNA, though, are that at one end it carries the amino acid, okay? The amino acid, which is the part of the protein, the building block for the protein. The other end is going to have a sequence that matches or is complementary to the codon, which we'll find on our mRNA. Now, the codon is what codes for the specific amino acid. The anticodon, which is found on the tRNA, is the part of the tRNA that allows the molecule to form a bridge between the codon and the amino acid. So this tRNA knows that if it contains the anticodon GGC, that is complementary to the codon CCG, and so we would need to have up here the amino acid attached that corresponds to this codon, CCG. Okay, so the tRNA's two important parts are the anticodon and the amino acid that it carries. Okay, so let's look at this process of translation. It occurs in 3 steps as well, those same 3 steps: initiation, elongation, and termination. Translation is a little bit different than transcription, though, because again we're reading our mRNA in these codons that are 3 bases each. So in initiation, what we see is that the ribosome subunits, so remember the large subunit and the small subunit, come together, and the first tRNA, they're all going to bind to the mRNA molecule. And notice that it says that the first tRNA is always AUG. AUG is a codon that codes for methionine, and what actually ends up happening is that the ribosome attaches to the mRNA and it travels along until it finds the first methionine codon. And that's the point at which that first tRNA will bind, and we say that initiation is complete, because we're now ready to move into elongation, where we see a tRNA comes in to the next site. We form a bond between those 2 amino acids, and now the ribosome is going to move down. As the ribosome moves down, that first tRNA exits. It enters the E site and exits. A new tRNA enters the A site, forms a bond, moves down, a new tRNA enters, the first tRNA leaves, and so on. So we got a little bit, the animation got a little bit ahead of me, but essentially what's happening is that as the ribosome moves, we're going to see a shifting of those tRNAs. So the tRNA from the A site shifts to the P site, the tRNA from the P site shifts to the E site, and it leaves. And that leaves the A site open, where it is positioned now over the next codon, so that allows for the next tRNA to enter, bringing with it an amino acid, and we form a bond. It moves down again, the next tRNA enters, brings an amino acid, and we form a bond. And that continues to happen until the tRNA, or until the ribosome encounters what we call a stop codon. So if you were to go back and look, and we can do that very quickly, go back and look at this table, you can see that UAA, UAG, and UGA all code for stop codons. Those are the signals that the ribosome needs in order to say, okay, it's time to stop translation. And when it encounters one of those signals, the ribosome releases the protein that has been made, and it falls off of the mRNA. Okay, that allows the mRNA now to go on and be translated by another ribosome and make more protein. And so this is just showing us a very simple protein that's been made. Okay, so let's translate an action, and this is a little bit cheesy, so bear with me. When we do it in class, it's a little more fun. But what we see here is an mRNA sequence, and so if we want to translate that into protein, the first thing we want to do is identify our codons. So our codons are UCC, AUG, AUA, CUU, GAA. Now remember, we would always start with the AUG. However, for my little fun example, we need a different letter at the beginning, and so we're going to start with UCC, but this is typically where our protein would start. So refer to our table to find the amino acid that's identified or defined by each codon. So our first codon is UCC. So we're going to find the amino acid that corresponds to UCC and write that in our protein sequence. So U is my first base, C is my second base, and C is my third base. So UCC codes for serine, and the one-letter code for serine is S. So our first letter is S. Our next codon is AUG. So if we go to the table, our first base is A, our second base is U, and our third base is G. So methionine is the amino acid, which is also known as the start codon, and it has a one-letter symbol of M. So our next letter is M. AUA is our next codon. So A, U, A. So our next amino acid is isoleucine, which has a one-letter code of I. So our next amino acid would be I. Our next codon is CUU. So CUU. So leucine is our next amino acid, which has a one-letter code of L. I'm sure you see where we're going. So L is our next amino acid. And then our final codon is GAA. And if we go to our chart, G is our first base, A is our second base, and A is our third base, which codes for the amino acid glutamic acid, which has a one-letter code of E. And so now we can smile because we understand how translation occurs. Okay, so that's the process of transcription and translation. I like to use a very common example, something that you're familiar with, to relate things to whenever we're looking at a complex process, and I think that most people can relate to going out for a meal. And so we're going to look at how going out to dine in a restaurant might compare to the process of transcription and translation. So if we look at transcription, you can compare this to actually going into a restaurant. When you sit down in the dining room of that restaurant, you're actually in the nucleus because you're given a menu. And the menu tells you all of the possible things that could be made by the kitchen. And so that would be kind of like all the possible proteins. So this is really the genome of our restaurant. The genome of your cell is all the genes that that cell contains. Now, when you place an order, you're telling your waiter to make a specific gene, to make that specific gene into a protein. And so when your waiter takes that order down, writes it down, he's going through the process of transcription, taking your verbal order and writing it down into written form. And then that produces your mRNA. Okay, you may go through the process of mRNA processing because you might tell the waiter, you know what, hold the pickles, or I'd like my taco salad with no sour cream, or my chicken salad with dressing on the side. Okay, so you're providing some modifications to the original order. Okay, so that's transcription and mRNA processing. And what's going to happen is that that waiter is then going to take the order into the kitchen, which would be our cytoplasm of the cell, and is going to provide the order to the chef. Okay, so the order itself, we said, is the mRNA that has been processed. Pardon me, that mRNA that has been processed. And when we give that order to the chef is acting as the ribosome. Okay, and the process of the ribosome working with the mRNA to prepare our protein, or the chef preparing the order, is translation. And then the final product, the prepared dish, would be the protein that's made at the end of translation. Okay, so it's very easy to take this process and correlate it to something that you're familiar with to help you remember the steps. Okay, so we're going to wrap up with just a few kind of loose ends, things that we need to know about transcription and translation outside of what we've already discussed. And one of those is that we don't typically see a single ribosome interacting with a single mRNA all by itself. Typically, you're going to have multiple ribosomes that attach to your mRNA. So for example, this ribosome, or this one even, was the first one to attach, and it moved along and made its protein, and then when it gets to the end of the mRNA, or the termination sequence, it's then released and it breaks apart. This ribosome was second and it moved along. This one was third, this one was fourth, this one was fifth, and this is the one that's getting ready to attach to the mRNA. And those ribosomes just move along and make protein, and it allows us to make a maximum amount of protein from a single mRNA because we're making it, every ribosome is working on it all at the same time, but they just may be at different stages of building that protein. So we call that a polyribosome. Okay, just like our mRNA went through the process of being modified, very often we see that proteins go through that process of being modified as well. And this is one of those things that we've talked about very briefly in previous lessons, but remember we said that in the Golgi apparatus, the proteins that are made by the rough ER can be modified. So they may fold into a particular shape, they may have things added to them like sugars, lipids, or phosphate groups, and in some cases we may see that the protein gets cleaved or part of it gets broken off. And insulin is a very good example of where this happens, where here's our long chain of insulin and it kind of folds in on itself so that we can form disulfide bridges between the alpha chain and the beta chain, and then all the rest of this gets cut off so that what we're left with is just these two parts of the insulin protein, but those two parts are what we need to do the job. Another thing that we need to know about translation is that there is something called a reading frame, which is important. So this was our protein that we translated earlier, and it's based on the codons that we saw here: UCC, AUG, AUA, CUU, GAA. But what we have to recognize is that there's really multiple ways to read this. What if I started with the C instead of the U, or the second C instead of the U? That would give me a whole different set of amino acids. So if we started with the C, we would actually have a proline and then a stop codon. Or if we started with the second C, we would actually have a histidine, a couple of other amino acids, and then a stop codon. Okay, which gives us a completely different protein than SMILE. So the reading frame ends up being important when we have things like deletions. Deletions of one or two bases can cause what we call a frame shift mutation, where we see this would be a 1 base pair deletion or a 2 base pair deletion that causes our reading frame to shift so that it's read in a different amino acid sequence. However, if we were to have a 3 base pair deletion, we wouldn't see a shift in how the reading frame was read. We would just see a deletion of this whole amino acid, but we would still get MILE. We may not get SMILE, but we would still get MILE. Okay, so those are going to give you varying degrees of severity when it comes to problems with the protein that results at the end. Some other types of mutations that we can see having an effect on our proteins. Missense mutations would be those that cause a change in amino acid sequence, and nonsense would be like what we just looked at, where we have an early stop codon. So this would be a nonsense mutation because we now have a protein that's only 1 amino acid long, and obviously that's not going to be terribly effective. However, if we had a change in our AUG that turned that methionine into an isoleucine, so maybe from AUG into AUA, then we would have SIILE. That would be a missense mutation because it changes 1 amino acid in the sequence. We can also have things called silent mutations, and so if we go back and look at a chart from earlier, if you notice, for example, proline, threonine, alanine, we talked about these a little bit earlier, that last base is identical. We said there's redundancy there. Well, if we have a mutation that changes ACU to ACC, we still get a threonine amino acid. Or GCU to GCA, we still get alanine. So those mutations would be considered silent mutations because they don't actually have an impact on the protein that gets produced. They have an impact on the DNA sequence, but that DNA sequence is only there to create a protein sequence, and so it really does not have a significant impact. Okay, that's all for our chapter on transcription and translation. Our next chapter will be a very, very brief look at gene expression regulation in chapter 18.